we have concluded our previous lecture, what I personally consider to be the question. How come that an egg cell and a sperm cell combine and give rise to a whole organism? In our case, we have over 200 different cell types. And when we say different cell types, we are talking really different. We have skin cells, we have bone cells, we have blood cells. Those are really different cell types. So we know that there is about 20,000 genes in our genome. And we also know that a fraction of them are expressed in each cell type. So intuitively, if we would pack the genomic DNA such that we can deny or enable access, that already right there would present means to regulate gene expression. One very nice example are the sex chromosomes. So it is commonly depicted that the woman has the letter XX and the man XY. But when you look at the actual chromosomes, then you see that they are clearly very different. And 2009, the X chromosome has been then sequenced. And since then, we know that we have about 150 million base pairs and up to 2,000 genes. So there is a lot of information. Y chromosome, on the other hand, is tenfold smaller and maybe maximum 200 genes. So that is a huge difference. The question is now whether or not there is really a male disadvantage. Let's say we have a gene A on the X chromosome. In the female case, when this gene expresses, we are going to have a gene product twice as much as in the male case. And this would be a quite imbalance. And what nature does in this case is to randomly take out one of these two X chromosomes through inactivation. And this is indeed been done through chromosome packaging. Another question is, of course, is it the way how nature deals with this problem, like bringing justice between male and female? When we look at our not really close relatives, Drosophila, in this case, instead of taking out the X chromosome, in the female case, it upregulates the X chromosome twofold in the male case. So it seems nature is really very versatile. We now take a look at a typical nucleus. So what do we see? So this really looks like a Milka coup in black and white. What are we looking at? So these dark, compact areas are the heterochromatin, the inactive regions of the genome, and we have the open euchromatin, U like eukaryote, the true chromatin, that is the active one. Now the big question is, why do I see a structure like this? We definitely know that the DNA is wrapped around the nucleosome. So how come that the cell or the nucleus end up looking like this? And as I announced in the previous lecture, we have a brand new concept, but also an extremely intuitive concept. If you have ever made a salad, then you know that oil and vinegar will not mix. This is basically how you can intuitively imagine what the DNA material does in the nucleus. Is it really like oil and vinegar? Let's do the experiment and see. To this, again, we go back to our reconstitution experiments and make a beads on a string. So we wrap the DNA around the nucleosomes, create a polynucleosome, and then ask whether or not we can form these droplets. And indeed, if you increase the magnesium concentration, for instance, you start seeing these spherical shapes. Or if you do increase the concentration of these polynucleosomes, you also start seeing these round shapes. Well, the question is now, of course, how do I know that they are not some aggregates that just appear? Well, this you can then also watch. Here you see these liquid droplets fusing. If they were aggregates, they would just sit on the cover slip and do nothing. And indeed, with this, we can then say the so-called liquid-liquid phase separation properties are indeed coded in this molecule. So there is a concentration dependence, there is a dependence on ambient conditions. So I can change the conditions and in order to see this effect. And they are indeed liquid, because if they weren't, I wouldn't see this fusion events. So we can then say that chromatin indeed phase separates. Phase separation is in fact not only relevant for the inner life of the nucleus, but the whole cell. So within a eukaryotic cell, you probably know many membrane-bound organelles, like mitochondria, the fuel machine that makes all the ATP, or the tongue twister and the plasmic reticulum that docks to the nucleus in order to get the RNA for protein synthesis, etc., etc. But you are probably less familiar with the non-membrane-bound organelles, like P-body, U-body, whatever that means. So the concept is that the cell uses phase separation as a mean to organize the whole non-membrane bound regions of the cell. In the next couple of slides, we are going to look at the major aspects of phase separation. So these slides are completely inspired by the iBiology lectures given by Cliff Brangwine. I highly recommend these lectures. So he is, as opposed to me, a physicist and has a brilliant concept to explain what phase separation is. The basic question here is, what are the conditions of mixing versus demixing? So when we look at a picture like this looks familiar, intuitive. So when I mix two different things and wait, they are going to mix up completely and get to the highest entropy possible. The other question is whether or not we can go back. Yes, we can go back, but this of course depends on the physical characteristics of these balls, right? So this now brings us back to our oil and vinegar. So you can shake it up, so get it, if you will, to the high entropy, wait, that one will go back to the low entropy and separate. In this concept, it turns out that the number of interactions that these respective balls can initiate is very important, whether or not at the end they would phase separate. So if we have just one interaction interface, then we are going to have a dimer. But if we then introduce another valency, then we also increase the likelihood that they interact with each other. And I keep doing this, then I can have the third valency, and then they are going to start making more and more contacts, and this ends up being the most important variable whether or not you are going to have a phase separation or not. What are these high valency interactions in a given protein? Well, it turns out that these so-called intrinsically disordered regions are very important in order to observe phase separation. These are encountered often, so up to 30% of the proteome are the so-called IDRs. They are floppy, can adopt many conformations, and with that they can also initiate different types of interactions and achieve this multivalency in order to phase separate. In the meantime, we have many examples of phase separating proteins and their properties, etc. But the other question is also, why is it important to understand the phase separation properties of proteins, DNAs, RNAs, etc.? Well, it turns out that there is a link between the phase separation properties of a protein and disease. So I will show you one example, and you definitely know this disease. So it is about this Fas protein. Doesn't matter where this cryptic name again comes from, but this is a protein that is found in the nucleus, and this is a DNA RNA binding protein. This protein contains these intrinsically disordered regions and is known to form these liquid droplets up to jellification upon stress, for instance. But as long as these phase separation properties are regulated like this is not a problem. The problem is if these properties then are changed and that the phase separation ends up being an irreversible aggregation. And this property, you know, from the ALS disease that the famous physicist Stephen Hawking had, the late Stephen Hawking, unfortunately. And the question is now, of course, why are we seeing this, what we are seeing on the slide? And recently we also got an answer to this question. We have recently got an explanation for this observed behavior of this Fas protein. And as you can imagine, that it is important to really regulate the solubility of Fas, because if the liquid-liquid phase separation properties are changed such that it then aggregates irreversibly, this is exactly the disease-causing effect. And this indeed happens when this solubility factor is disrupted, which then eventually gives us this irreversible phase separation. Since we are already in the brain, let me show you another neat example. You probably remember these microtubule robes. So these are the filaments that would pull apart the replicated chromosomes during the cell division. And microtubules indeed were first discovered in these mitotic spindles, but later it turned out that microtubules are way more abundant in the brain, and this is indeed the reason why we can turn to pig brains and purify this protein out of this tissue, because they are that abundant. And this protein has many associated factors, one of which is tau. Again, don't worry about the cryptic name, but important thing here is tau has also these famous intrinsically disordered regions and has many microtubule binding repeats. So again, this multivalency. And what people have observed, and yes, tau with its intrinsically disordered regions turned out to also be able to do this phase separation and form these liquid droplets. And what people observed is when these tau droplets were around, they have been able to observe microtubule polymerization. So this was basically a nucleator for the microtubule polymerization. And if you would not have any tau in the solution, you would not observe any polymerization whatsoever. So this short excursion was another nice example of the interdisciplinarity of life sciences. In the next lecture, we will go back to DNA and its processing, and in particular, we will look at E. coli, how it assembles an amazing machinery in order to replicate its DNA.